Hello, everyone, and thank you for joining today. My name is Lea Vala, and I will be your moderator for this webinar, and you will learn on Prometheus Panta and how this platform supports your biologics developability assessment with 4 integrated biophysical technologies for stability characterization. It's my great pleasure to introduce our speaker today, Nathan Wallace. After his PhD in biology at the University of Dayton, Nathan did a postdoc on HIV and joined Nanotemper Technologies. Currently, he is supporting our customer in the Midwest of the United States with their projects on biologics characterization. Nathan, the stage is yours.
Thank you so much, Leah, for that kind introduction. As she said, my name is Nathan. I'm in FAS supporting the Midwest of the United States, and today I'm super excited to be sharing with you how you can take advantage of our Prometheus Panta instrument to help improve the developability of your biologics. Now, before we get started, I always like to start off every presentation with a brief agenda, just so you have an idea of what we're going to try to cover today. So first off, we're going to talk about developability and why it's important to have that early on in your biologics workflow. We'll then talk about which of those parameters our Prometheus Panta instrument can measure for you. And then we'll do a brief introduction in terms of the technologies that live within the Prometheus Panta. There are 4 of them. And then once we understand the technology a little bit better, we'll give you a real-life example, a case study that we developed with the Merck company. We'll talk about what criteria Merck uses for developability, go over their experimental design, and talk about what they learned, have some conclusions, and then we'll spend a little bit of time at the end wrapping up, and as Leah said, leave plenty of times for questions. So early on in your development and formulation process, typically you have very little sample to work with, and from that you need to learn a multitude of information from it. Whether you're screening different constructs or looking at different buffer conditions, it's critically important to ensure you understand as much about your sample prior to advancing it through your pipeline. Now, developability assessment can help mitigate the risk that is associated with selecting the wrong candidate or the wrong condition, which can save you in not only time, but also money. If you're able to eliminate those bad candidates early in the process, that's much more preferable to investing your resources into something that's never actually going to make it to market. Now, again, with those sample limitations early on in the development process, again, we're talking about those early screening stages where you're looking at different constructs or different buffer formulation. We want to learn as much as possible from just a small amount of sample. Now, Nanotemper's Prometheus Panta is a single instrument that's using only 10 microliters of your sample to provide many of the conformational and colloidal stability parameters that are required as part of a robust developability assessment checklist. Now, as I mentioned before, the Prometheus Panta has 4 technologies. We have NanoDSF, back reflection, and both dynamic and static light scattering capabilities. All 4 of these technologies together can help you build a more complete developability profile for your candidate. The data that we're generating is very high resolution with excellent data quality, which in turn is going to help reduce the amount of time and effort spent on interpreting results, and it makes it a lot easier to rank the desirability of your candidates for further development. So now that we've talked a little bit about developability assessments, I want to spend some time breaking down the technologies in the Prometheus Panta and how they can help you with those assessments. So as I'm sure we all know, proteins are incredibly complex macromolecules which behave equally as complex in solution. As evidenced by this slide, we can use all of the technologies in the Prometheus Panta to help develop a more thorough understanding of not only conformational, but also colloidal stability of your molecules. And the beauty of this is we're doing it simultaneously during a single thermal ramp with only 10 microliters of your sample. Now, the Prometheus Panta gets its name from the Greek word panta, which means all-encompassing. And this instrument does truly provide you with a plethora of information regarding your sample's conformational and colloidal stability. The first technology is NanoDSF, or nano differential scanning fluorimetry. What we're doing here is we're monitoring intrinsic fluorescence of aromatic amino acids in response to a thermal gradient. We also have dynamic light scattering, or DLS. This is going to provide you with information regarding the purity of your sample, as well as the actual size of your particles in solution. We also have our back reflection technology. This is going to give you information regarding the turbidity of your sample, which gives you an idea as to whether or not you have a large amorphous aggregates in your system. And finally, we have static light scattering, or SLS, which is another way to inform on colloidal stability. And you can actually set up additional experiments to give you information regarding the average molecular weight of your sample, or even a self-association parameter known as the B22, or the second virial coefficient. Now, again, NanoDSF, what we're monitoring in this case are changes in intrinsic fluorescence of those aromatic amino acids. We're doing this in response to either a temperature or a chemical denaturant. Okay? The actual wavelengths that we're monitoring are 330 and 350 nanometers. And as you can see in the chart on the left, those correspond very nicely to, at 330 nanometers, those embedded tryptophans, and at 350 nanometers, those surface exposed tryptophans. And so what we actually do is demonstrated on the right. We plot a ratio of the 350 divided by the 330 nanometers. And we plot that versus either the temperature or the concentration over chemical denaturant. And from that, what we're able to do is build these really nice unfolding curves from which you can derive useful parameters such as the TM at the inflection point of that sigmoidal unfolding curve. When you're looking at your NanoDSF data, the inflection point of unfolding can sometimes be subtle, especially in the case where you have multiple inflection points near one another. That's shown in the top graph. What we can do in the software very easily is actually transform that data into what we call the first derivative view. This makes it much easier to visualize where those inflection points occur, so it's a lot easier to determine your TM ranges at a quick glance. Our back reflection technology is going to look at the amount of light that gets attenuated by your sample. This is going to inform us on sample turbidity. Now, this can be really important because those larger amorphous aggregates can be potentially toxic for patients once your samples actually make it to the clinic. Now, dynamic light scattering, or DLS, it's going to look at the light that your particles in solution scatter at a given angle. Now, your larger particles are going to move much slower than your smaller particles. Therefore, there are less fluctuations in scattered light that actually reach our detector. By processing those fluctuations, or the movement of those particles in solution, we're able to use that information to determine not only the size, but also whether or not you have multiple different sized populations in your sample. Now, static light scattering, or SLS, is also looking at the scattering intensity of your light from your sample. However, unlike DLS, we're looking at the average scattering of all of your particles in the sample, even if it's a mix of aggregates as well as folded protein. Now, increases in scattering, generally speaking, indicate the presence of larger particles or aggregates. And again, as I said before, you can also set up subsequent experiments where you can calculate the average molecular weight of your sample. The great thing about all of this is that we're running those fluorescence optics in parallel with our light scattering optics. So you're collecting not only the thermal and colloidal stability data throughout the entire thermal ramp, and we're doing that on just the same 10 microliters of sample. The benefit of adding those size changes throughout the thermal denaturation ramp is that it gives you more information about the actual mechanism of unfolding and aggregation, including questions about whether or not those large irreversible aggregates are forming, or if you just have some smaller order oligomers beginning to form. So now that we've spent a bit of time talking about the technology that lives within the Prometheus Panta, let's take a look at a real-life example. So Merck and company have been enthusiastic users of our older model of the instrument called the Prometheus. This instrument just had the NanoDSF and back reflection technologies. However, a few years ago, they actually upgraded to a Prometheus Panta. So what I want to share with you now is by how adding DLS, it actually helped improve their developability assessments without requiring the need for additional experimentation or even additional material. So first off, I just want you all to take a look at all of the different measurements and parameters Merck is taking into consideration during their developability assessment. As you can see, based off of this list, there are a ton of quality attributes that are considered when they're selecting those best candidates to move forward for further development. Now you can look at the exact same list, except highlighted in blue now are all of the categories that the Prometheus Panta can measure for them. Now it's worth noting that there's not one instrument that exists that can measure everything. But if you're able to consolidate some of those assessment steps, you're able to save time and material for those other parts of the assessment process. In Merck's case, Panta is particularly critical for their stability assessments. It's able to answer questions such as, how stable is my molecule? Is it unfolding? Is it aggregating? Are there any contaminants in our solution? How will it remain stable long term, say at room temperature? Or how do changes to the primary sequence or the buffer conditions actually impact those stability attributes? All of these questions can be answered using the Prometheus Panta. So for today's case study, we will be specifically addressing monoclonal antibodies. So as you can see here is a representative model of how those monoclonal antibodies behave in response to a temperature gradient. It probably looks pretty similar to the one that I showed you before. The main takeaway from this slide is that there's always an interplay between how well folded a protein is, whether or not that process is potentially reversible, or if you get to the end point where you have complete irreversible aggregation. Now, thermal stability assessment, in this case, is done with NanoDSF. Again, this is providing us with information about the conformational stability of our monoclonal antibody. And just a quick reminder, NanoDSF is using the intrinsic fluorescence of aromatic amino acids to monitor how well folded a protein is. And it's really awesome because we have such high quality data on the Prometheus Panta, you're able to actually see some of these unfolding transitions down to the domain level. Now, NanoDSF is going to provide you with some useful parameters, such as the TM, or the melting temperature, and the T onset, or the onset of melting. And you can use these parameters to help rank or triage your candidates. Now, again, generally speaking, higher values for the TM and the T onset generally reflect a more conformationally stable monoclonal antibody in this case. Now, with the addition of those light scattering optics, we also get information about the purity and the colloidal stability of a sample. The Prometheus Panta performs DLS measurements in parallel with the NanoDSF thermal unfolding assessment. This means that you measure the size of the particles in solution as well as the PDI. It's a measurement of how well folded a protein is and whether or not there are different protein species present in that sample. We can take those alongside the melting temperature and the onset of melting parameters as well. Another cool parameter that is provided from DLS is something called the T scattering. What this is a measurement of the temperature at which the particle starts to change size. And this is really interesting because it's an additional indicator of aggregation or instability that is completely independent from the conformational stability information gained from the NanoDSF. Now, in this particular case, Merck's goal was to try and optimize the biophysical properties of an originator monoclonal antibody. What they did from there is they made some rational changes to those originators to make the parent monoclonal antibody generation more stable. Again, these were modifications that were known to confer some stability advantages. They then took those parent monoclonal antibodies and made an additional 2 to 3 point mutations in a certain region of that monoclonal antibody. And they did this systemically to each of the daughter antibodies. And what I mean by that is each of the shades of color correspond to the same 2 to 3 mutations as one another. From here, the goal was to determine how these mutations actually impacted the stability compared to the parent antibodies. They collected stability data on each of these constructs using the Prometheus Panta, and then they ranked those candidates based on the melting temperature, the onset of melting, the onset of turbidity, the T scattering, and the polydispersity index. Ultimately, the goal was to try to avoid suboptimal properties and determine if there were any trends that could make a candidate more ideal for further development. So now we'll take a look at some data. First, we're comparing the data the thermal stability and the colloidal stability of the parent monoclonal antibodies. You can see that even though the parent monoclonal antibodies had only modest primary sequence changes, this resulted in dramatically different profiles. So we already know from this that just small changes to the sequence can have a big impact on the stability of a monoclonal antibody. I want to take a closer look at protein A, shown here in black, which we see has the lowest TM, indicating that we have some conformational instability, and it also shows a dramatic onset of turbidity and changes in the cumulate radius, indicating that we have some colloidal instability as well. Now, protein A and its daughters are shown here. So protein A is still shown in black, and the daughters are all shown in blue. Here, even though we have even fewer changes to the primary sequence, again, we're talking about only 2 to 3 point mutations per construct, you're still seeing big changes in the profiles. The melting temperatures are all decreased compared to the parents. However, the scattering and the turbidity data actually show some improvements in terms of colloidal stability. So now if you're like me and you like to see things in numerical form, I provided that for you here. It's worth noting that if you look, there's not a single construct that has the best statistics across the board. The actual selection of those optimal candidates is always going to be a judgment call by you, the researcher, where you have to decide which parameters are most critical for decision making or which ones are cause for immediately eliminating a candidate. Here you can see that the changes to the TM are fairly modest, but the high PDI of constructs 2 and constructs 4 are a red flag for colloidal stability and probably not constructs that you want to move forward with. So you may be asking yourself at this point, why is it important to have both NanoDSF and DLS data? So with NanoDSF alone, it can become a little bit harder to triage or stratify your constructs. So here we can see that of all the constructs evaluated, they were ranked based on their TM and their T onset. That is, all of the data that's related to conformational stability or when is the protein actually unfolding. So as you can see here, over half of the constructs have acceptable values for these parameters, which isn't really helpful for you when you're trying to rank and select those optimal candidates to move forward. When we add in the information from the DLS, we're now evaluating the exact same samples on 2 different physical properties. And as you can see, a few of those most ideal candidates really pop out here. We have a high unfolding temperatures and virtually no scattering at all. That is, that there's no aggregation associated with those constructs, which again is often a positive sign for that long-term storage. So I just want to take a moment now to sort of wrap up and take a few key takeaways from this case study. So I hope that you have understood the importance of developability assessments and all of the different biophysical attributes that Merck takes into consideration when pushing a candidate through their pipeline. So hopefully able to show you that the Prometheus Panta is able to measure many of those stability attributes in parallel during the same thermal ramp using only 10 microliters of sample. Able to take both the NanoDSF and DLS data together to get a more complete picture of the conformational and colloidal stability of your molecules. And if you're able to measure multiple of those stability attributes on the same sample, it's going to make it a little bit easier to rank your candidates and triage them accordingly. So now, I'd like to wrap up by elaborating a bit more on what the Prometheus Panta brings to the table when it comes to your developability profiling. We discussed with the case study how the Prometheus Panta is able to help with the initial triage by identifying the impact of certain mutational changes have on a rationally designed construct, or you could even apply this to randomized libraries as well. We also show how the resolution of the NanoDSF data in particular can help unveil exactly how those candidates are unfolding. There are other developability attributes that light scattering optics help reveal, including long-term stability from accelerated stability studies, and self-interaction measurements that can help you predict how your samples may behave at high concentrations that are typically required for clinical administration. So in summary, the Prometheus Panta provides you with a ton of information regarding your candidates and the different conditions that you'd like to test them in without requiring very much material or running multiple experiments. Now, you can run over 1,500 samples completely hands-off with our robotic autosampler upgrade, or you can run just a few at a time, so the throughput is completely flexible to your needs. The high resolution data we're generating makes it super easy to identify trends and draw conclusions about your sample. And the data quality is so high, it can help you stratify those constructs even with just modest changes to the TM, which is only going to help improve your selection criteria. And with that, I would just like to thank everyone for their time and attention.